A number came back from a genome sequencer in a clean room at Harvard Medical School, and the researchers ran it again, not because of excitement, because it didn't fit. The paternal line said one thing. The maternal line said something else entirely. And the ancient samples pulled from teeth extracted in a parking garage excavation in central Germany said a third thing that made the first two harder to reconcile, not easier. All three results were real. All three were published. All three are still being argued over. This is the data scientists are most careful about sharing, not because it is wrong, but because it is incomplete in a way that no one has yet found the words to explain cleanly. For most of recorded history, the origin story of the Jewish people has been one of the most documented human narratives on Earth. The lineage begins in the ancient Levant, in the Iron Age hill country of Canaan, in the kingdoms of Israel and Judah, in a region that corresponds roughly to modern-day Israel, the Palestinian territories, Lebanon, and parts of Syria and Jordan. This is not myth. This is not religious claim, dressed as history. It is a story that archaeology, linguistics, and population genetics have all, in their different ways, confirmed. The first major rupture came in 586 BCE, when Nebuchadnezzar II of Babylon conquered Jerusalem, destroyed the first temple, and deported a substantial portion of the Judean population eastward. The Babylonian exiles scattered a people who had, until that point, been geographically anchored. They returned, partially. They rebuilt, partially. Then, in 70 CE, Rome destroyed the Second Temple and began what would become nearly 2 millennia of dispersal east into Mesopotamia and Persia, west into the Mediterranean, north into the European continent. Out of this dispersal came 3 broad communities that geneticists and historians track today. The Ashkenazi Jews, who eventually settled in Central and Eastern Europe and developed a distinct culture and language, Yiddish. The Sephardim Jews, who built communities across the Iberian Peninsula, expelled from Spain in 1492 and dispersed across the Mediterranean and North Africa. And the Mizrahim Jews, who remained in or near the Middle East, in Iraq, Iran, Yemen, Egypt, whose connection to the Levantine homeland was never geographically interrupted. 3 branches. 1 described origin. And the Y chromosome, think of it as a kind of surname passed down not in ink but in the body itself, handed from fathers to sons with almost no alteration across generations. The Y chromosome told a consistent story. More than 70% of Jewish men across these 3 communities shared paternal lineages that clustered with populations from the northern Fertile Crescent, Lebanese, Druze, Syrians, Samaritans. The genetic signature of ancient Levantine men, preserved across 2,000 years of diaspora, across expulsions and massacres and forced migrations, the fathers had held their line. The story was clean. The data supported it. For decades, this was what the science said. The story holds for the fathers. When scientists looked at the mothers, the map changed entirely. Here is where it gets complicated. Mitochondrial DNA is the other kind of inheritance, the maternal line, passed exclusively from mothers to children of any sex, unchanged. It is the counterpart to the Y chromosome. If the Y chromosome is the paternal surname written in biology, then mitochondrial DNA is the maternal surname. And in a population where both lineages are ancient and stable, you would expect both to point in the same direction. They do not. In 2013, a research team led by Martin B. Richards of the University of Huddersfield, working with collaborators from the University of Porto and other institutions, published a study in Nature Communications that examined mitochondrial DNA across Ashkenazi Jewish populations. The Ashkenazim carry a distinctive mitochondrial pool, 4 major founding lineages that are rare or absent in surrounding European populations, and which had previously been assumed to trace back to the Levant. Richards and his team looked at those 4 lineages with higher resolution than any previous study had managed. The result was not what anyone expected. All 4 major maternal founders accounting for roughly 40% of Ashkenazi mitochondrial DNA variation, did not originate in the Near East. They did not originate in the Caucasus, as the Khazar theory had sometimes suggested. They had ancestry in prehistoric Europe, not Roman Europe, not medieval Europe. Prehistoric Europe populations that were already old on the continent before the word diaspora existed. And when the researchers extended their analysis to the remaining minor maternal lineages, those too pointed predominantly toward Europe. Their conclusion: approximately 80% of Ashkenazi maternal ancestry comes from women, indigenous to Europe. 8% traces to the Near East. The rest is uncertain. Not by a little, not by years, by millennia. What this means, in plain language, is a phenomenon geneticists call sex-biased admixture, and it is exactly what it sounds like. Levantine men moved into Europe. The women they had children with were local Europeans, not women brought from the Levant. The religion passed through the fathers. The covenant passed through the fathers. The identity passed through the fathers. But DNA passes through the body, not through choice, and the bodies of the mothers said something the fathers' stories had never mentioned. The result was published. The data has been replicated in subsequent analyses. This is not a fringe finding. It is one of the more cited results in the archaeogenomics of European Jewish populations. But it left the central question entirely open. If 80% of the maternal line is European and the paternal line is Levantine, then what exactly is the genetic composition of the Ashkenazi Jewish population? And who were the women? In 2013, a decade after the paper, archaeologists digging under a parking garage in Erfurt, Germany, found the beginning of an answer and an entirely new set of questions. The first theory is the oldest one, and it still has genuine scientific weight. It is called the Rhineland hypothesis, and it works like this. Jewish men moved westward out of the Levant in waves after Babylon, after Rome traveling through Alexandria, through the Greek islands, through the Italian peninsula. As they moved, they settled, traded, married. The communities that formed along the Mediterranean coast of Italy, particularly in the south, became the base population from which Ashkenazi Jews eventually emerged. From the Italian communities, families moved northward, following trade routes through France and into the Rhineland region of western Germany, where the first formally documented Ashkenazi communities appear in the historical record around the 10th century CE. This theory received remarkable support from the 2022 ancient DNA study. A team led by Shai Carmi of the Hebrew University of Jerusalem and David Reich of Harvard Medical School, working with the Max Planck Institute for Evolutionary Anthropology in Leipzig, extracted genomes from 33 individuals buried in a 14th century Jewish cemetery in Erfurt, Germany, the largest ancient Jewish DNA study ever conducted. The cemetery had been uncovered during a rescue excavation in 2013, when construction workers converting a 15th century granary into a parking garage found graves beneath the foundation. The Erfurt results supported the Rhineland framework in a striking way. The ancient individuals were genetically very similar to modern Ashkenazi Jews, implying that the Ashkenazi gene pool has been remarkably stable since at least the 14th century. And ancestry modeling consistently pointed toward Mediterranean Southern Europe people related to modern South Italians as the dominant pre-medieval source. The bottleneck signal was equally striking. One specific mitochondrial variant, found in 20% of modern Ashkenazi Jews and nearly absent in non-Jewish populations, appeared in 35% of the Erfurt individuals. A third of the cemetery, 35%, descended through their maternal line from a single ancestral woman. The founding population, by this estimate, was probably no larger than 500 people at its core. The Rhineland hypothesis explains the paternal Levantine signal. It explains the Italian corridor. It explains why Ashkenazi Jews show specific genetic similarities to modern populations in southern Italy and Greece. It is, by any standard, a serious and well-evidenced model. Its limitation is this: if the European women who joined these communities were primarily from the Mediterranean, from Rome, from the Greek coast, from the southern Italian towns, along the Rhineland migration route, then the maternal DNA should cluster there. It should look Italian. It should look Roman-era Mediterranean. It does not. It traces to women from prehistoric Europe, from populations that predate the Roman Empire by thousands of years, populations that were already deeply established across the continent before any recorded Jewish migration into it. The Rhineland hypothesis does not explain that breadth. It does not explain the depth. This is the part the records don't explain. The second theory is more complicated, and it requires looking not at where the Jewish communities eventually settled, but at the Roman world they moved through on the way there. In the early centuries of the Common Era, roughly between 100 BCE and 500 CE, Judaism was a legally protected, actively proselytizing religion within the Roman Empire. This is documented. Josephus wrote about it. Roman writers complained about it. At certain points, conversion to Judaism was not a fringe event, but a recognized social pathway, particularly for women entering Jewish households. Communities across Italy, Greece, North Africa, and the broader Mediterranean did not merely tolerate Jewish presence, they absorbed it, married into it, converted into it, at a scale that mainstream history has consistently underestimated. The second theory proposes that this is where the maternal lineages come from, not from women picked up incidentally along a medieval migration route, but from a systematic absorption of local women across the entire Roman Mediterranean over centuries before the community ever moved north. Women from Greek trading families, women from North African coastal towns, women from Italian agricultural communities whose genetic lineages traced back not to the Near East, not to Rome, but to the prehistoric farming populations that had settled Europe thousands of years earlier. This theory is consistent with the Erfurt data. The study's ancestry modeling found that the primary source of pre-medieval Ashkenazi ancestry was related to people living today in Mediterranean, Southern Europe, specifically South Italians, which is exactly what you would expect if the conversion event happened during the Roman-era Mediterranean phase before the northward migration. And the small founding population makes a different kind of sense under this model. Only 500 people. A community that had already been through its formation, already compressing the female European lineages and the male Levantine lineages into a single genetic profile before it ever reached the Rhineland. The bottleneck happened before the records start. The limitation of this theory is the same anomaly that limits the first. If conversion happened primarily in the Mediterranean, why does the maternal signal reach into lineages that predate Rome entirely? The prehistoric European women in the mtDNA pool were not Roman converts. They were carrying lineages that were already thousands of years old when Caesar was born. Something is reaching further back than either model currently accounts for. The third possibility is the one most researchers are hesitant to say out loud, not because it is disreputable, but because it cannot yet be tested. The Costa 2013 study was careful with its language. The maternal founders have ancestry in prehistoric Europe. Prehistoric is not a casual word. It means before written records. It means the genetic signal is not from the Roman period, not from the early medieval period. It is from populations that were settled across Europe in the Neolithic or earlier. Farming communities and hunter-gatherer remnants whose lineages have been ancient on this continent since before any of the civilizations this story involves had formed. For a moment, set aside the Rhineland route. Set aside the Roman-era converts. Ask a harder question. What if the mixing event that produced the Ashkenazi maternal pool was not primarily a story of Jewish men absorbing European women, but of something older, something that happened in a place and at a time that no study has yet sampled? In 2012, a geneticist named Eran El-Haik, then at the University of Sheffield, published a paper in Genome Biology and Evolution proposing that Ashkenazi Jews descended primarily from the Khazars, a medieval Turkic empire in the Caucasus that converted to Judaism between the 7th and 9th centuries CE. He claimed that only 3% of Ashkenazi DNA linked to the Eastern Mediterranean and that the rest pointed toward the Caucasus and Iran. The paper attracted enormous attention. It was also comprehensively contested. A 2013 study in human biology by a team including Noah Rosenberg and Doron Behar demonstrated that El-Haik had used modern Armenians and Georgians as proxies for Khazars, a population that has left no confirmed modern descendants, and that the similarities he identified disappeared entirely when proper reference populations were used. Researchers are careful to frame this as a methodological failure, not a fringe conspiracy. El-Haik's analysis used the wrong proxies, and when you replace them with neutral models, the Khazar signal is gone. But the anomaly that generated the Khazar debate is still there. The maternal signal is still prehistoric European. The paternal signal is still ancient Levantine. The founding population of 500 people lived before the 14th century, possibly before the 13th, possibly before the 11th. The Erfurt team noted that the founder event may predate the Erfurt community by as much as a millennium, which places it in the early medieval period at the latest. And from that window, the 7th through 11th century CE, the actual formation period of the Ashkenazi community, almost no ancient Jewish DNA has ever been sampled. The data doesn't confirm the gap exists for a reason. It only fails to rule it out. You are standing at the edge of a city wall in Erfurt, Germany. The year is somewhere between 1310 and 1340. It is winter. The air carries wood smoke from the market district on the other side of the wall, and frost has settled into the mortar between the stones of the cemetery at your feet. You are a woman in this community. You were not born into it. You came from somewhere north of here, or west, the daughter of a family on a trade route, or a village that had contact with Jewish merchants who traveled the river roads. You converted. You learned the prayers in a language you did not grow up with, Aramaic and Hebrew woven into rituals that were already centuries old before your grandmother was born. You raised children inside these laws, inside these walls, inside an identity that was not the identity your mother had given you. You chose it, or it chose you. The distinction may not have felt important at the time. And when you died, you were buried here in this cemetery that a 15th century granary would one day be built on top of, that a parking garage would be built on top of that, until 2013, when construction workers found the graves underneath. Your teeth are still intact. In a clean room at Harvard Medical School, 700 years after your death, a researcher drilled into the root of one of those teeth and extracted powder from the cementum and dentin. The powder contained fragments of your mitochondrial DNA. Fragments of the maternal line you carried, not the line you converted into, not the line your husband belonged to, but the older line, the one that ran back through your mother and her mother, and further, into populations that had been on this continent since before any of the civilizations in this story existed. Whose story is encoded in those fragments? The story you were born with, or the story you chose? The data doesn't answer that. It only records both. Here is the twist, and it is not a comfortable one. The feature of Ashkenazi Jewish genetics that has attracted the most scientific attention, the bottleneck, the founder effect, the extraordinary relatedness of a population that spans multiple continents, is not primarily a story of what was done to this community. It is, in part, a story of how the community was formed. The founder event predated the Erfurt community by a century at minimum, and possibly by a millennium. This means that the population that survived the 1349 massacre in Erfurt, when anti-Semitic violence killed or expelled most of the Jewish community of the city, had already been through its genetic compression. The expulsions, the pogroms, the forced migrations that marked the next 5 centuries of European Jewish history, they occurred after the bottleneck, not during it. The people who endured those catastrophes were already genetically the descendants of 500 founding individuals, already carrying the markers of that compression in their cells. The name changed, the city changed, the language shifted, the date of expulsion varied from place to place, but the genetics were already fixed. And that compression carried a cost that no one at the time could have known about. The disease-causing mutations that appear at elevated frequency in modern Ashkenazi Jewish populations, BRCA, 1 mutations associated with breast and ovarian cancer, Tay-Sachs disease, Gaucher disease, a cluster of conditions that genetic counselors now test for routinely. These were carried into the founding population and spread as it expanded, not as a consequence of persecution, not as a result of the ghetto. As a consequence of smallness, the markers of genetic suffering were encoded before the historical suffering began. The second reframe is quieter and harder. History remembered the fathers. It remembered the patrilineage, the covenant passed through the male line, the identity defined through paternal descent according to traditional halakha. The Levantine men who carried the faith northward into Europe, their story is documented. Their haplogroups are tracked. Their connection to the ancient Levant is the foundation of every origin narrative. But David Reich's team found that 1/3 of the Erfurt community descended, through their maternal line, from a single woman. One woman. Not a matriarch named in any text, not a figure commemorated anywhere. A woman whose lineage traced back